Assalamu alaikum girls again. Now I'm going to start from where I have left. I was talking about enzymes as they are of protein in nature and they are not chained during the reaction. They stay over there. They are not converted into a different product. Then I talked about substrate and products. If you look at this diagram, the top one, enzymes have a specific shape, and enzymes are acting on a chemical to convert into something else. That chemical is called as substrate. I repeat, enzyme is acting on this chemical will be called as substrate. And after this, when the enzyme performs its function, it converts the substrate into product. Look at this diagram. This is an enzyme. It has a particular shape, which we call it as a binding site. Every enzyme has a specific shape, and every substrate has a specific shape. So the specific substrate comes and binds to the specific enzymes. Randomly binding nahi hoti. The specific shape of the substrate allows it to bind to a specific enzyme, and this is how they make enzyme substrate complex, which is shown in the middle. Acha, when this complex is formed, now the enzyme is doing its function to speed up the reaction and convert the substrate into products. Here in the last diagram. Look at the first diagram, it was something else and we called it as substrate, but in the last diagram, now we call it as product. And what enzyme is doing over here is to speed up the reaction, to convert the substrate into product quickly. If that reaction was happening without enzyme in 2 hours, enzyme is going to speed up the reaction 1,000 times. Maybe it will take 2 minutes or 3 minutes, but I'm giving you a layman's idea or a random idea. But I will discuss later on that enzyme can speed up the reaction 1,000 times, or maybe I think it was written as million times. Let's see what happens next. So whenever a reaction happens, it requires energy. Reaction does not start on its own. Whatever the reaction is happening in the world, whether in the non-living environment or in the living environment, it requires energy to start the reaction. And that minimum energy required to start a reaction is called as activation energy. I'll give you an example of burning of stove. Whenever at home we burn or ignite the stove, we have to ignite splint, matches की तीली जलानी पड़ती है या lighter जलाना पड़ता है. We have to ignite the lighter or the splint and show it to the gas. अच्छा, अगर हम lighter ना जलाएं और हम splint को burn ना करें, तो you have seen it, gas doesn't burn. Gas doesn't, ignite. It doesn't turn into fire. It will stay as a gas. So who is starting this reaction? Basically, it's a chemical reaction, which is happening on our stoves. We see it every day. So that reaction is happening because of showing that splint. I'm recording at 8 a.m. and he's awake. Because of that, I record. So, Hamza, आप इधर बैठ जाए और मामा को सुने. सुने? चलो. अच्छा. So we require energy to start a reaction. And that energy is basically we provided here by burning the splint. And it is quite less energy, and as a result, the gas burns. This is called as activation energy, and it is required in every reaction. But some reactions happen at room temperature, some reactions happen at 0 centigrade, which means they require quite less energy. And some other reactions occur at 100 centigrade or 140 centigrade or 400 centigrade, which means those reactions require a lot of energy to start the reaction. This activation energy acts as a barrier. It's a hindrance to start a reaction. I mean, you have to provide energy to start a reaction, which is actually a hindrance for the reaction to proceed. So what enzyme do? Enzyme is actually lowering that barrier. Enzyme is lowering that hindrance. Look at this diagram. It's a symbolic representation. It is not like that enzymes are like pushing the or rolling up the huge stone up the hill, and it's a symbolic representation that in every there's a person who is rolling up a huge stone uphill. You know, is going to spend a lot of energy to push that stone, and as soon as that person reaches the peak, that stone will roll over the hill on its own and break into pieces, right? What I'm trying to tell here is that in every reaction, you have to push that stone. You have to spend the energy to start a reaction. As soon as you reach the climax, the reaction proceeds on its own. The reactants will convert into product on its own. So whatever the reaction is happening in this world, activation energy is required. Look at the 5 point. The energy needed to start a chemical reaction is called activation energy. Every reaction in this world requires energy to start, and that energy can be less, can be too huge, can be anything. And what is the work of enzyme over here? To lower this demand, to lower the demand of activation energy. So the reaction will start quickly and convert into product. I will explain it later on. Let's move to the next slide. So I was telling you the same point, that enzymes lower this barrier. Enzyme lower this hindrance. Let me tell you in my own way. For example, there's a reaction which requires 1000 kilowatt, or I'm sorry, I should say kilojoules or joules, whatever the pronunciation is. Let's say a reaction is requiring 1000 kilojoules of energy to start it. It's a quite huge energy to start the reaction. This is 1000 kilojoule activation energy. But if I'm going to add enzymes into that environment or into that cell, that energy to start a reaction reduces to 200 joules, kilojoules. So how much the enzyme is lowering the demand of energy? Yani wahi reaction which is going to start on 1000 by consuming energy of 1000 kilojoules, the same reaction will happen by consuming only 200 kilojoules. Now the question arises, how it happens? How the enzymes are able to do this thing? I think I have mentioned it somewhere in the slide that I have left here, I think in this slide. Yes, look at this pink line. I'm leaving that last paragraph of page 107, which I will teach you at the end of the chapter because it is a bit complicated. So I teach that paragraph at the end of the chapter. So enzymes have some ways to lower that demand. We will discuss it later. So in the presence of enzyme, the reaction is proceeding at a faster rate. When a reaction is happening, instead of consuming 1000 kilojoules, enzymes start the reaction by consuming 200 kilojoules. So obviously the reaction will carry out on its own and convert into products. So what you call it? Speeding up the reaction. Speeding up in a very faster rate, and as a result, less time will be consumed. Less energy will be consumed, less time will be consumed, and you will be happy. So let's move forward. Especially in artificial environments, when you use catalyst, I mean, in non-living environment, you use catalyst. So rather than consuming 2 hours, you will consume 10 minutes. So you will be happy to use a catalyst, and you will pray for a catalyst. So let's look at this graph. It is mentioned in your book as well, in your course as well. This graph, kindly look at the x-axis and y-axis. Do not say me, ma'am, samajh nahi aaya. You can ask question, obviously, but it is like a live lecture in the class. So just listen to me carefully that I'm talking about x-axis and I'm talking about y-axis. On x-axis, we are talking about progress of reaction, which means in the beginning of the x-axis, there were reactants. As soon as the time goes on, the reactants are converting into products. But here, the reactants are mentioned as substrate. So what is the progress of reaction on x-axis? Substrate is converting into product. And on y-axis, energy is mentioned. So as soon as, look at the pink line, as soon as the reaction start, energy is increasing. Someone is providing energy. Energy is increasing, and it reaches to peak, and then the energy lowers, and converts. Substrate is converted into product. Now this is the graph which has same pathway for every reaction in the world. Only the difference is कि peak different होती है. इसी तरह ही reaction का graph बनता है, energy का graph. कि first the reaction consumes energy, which is activation energy, and as soon as the demand is fulfilled, the reactants converts into products automatically, and energy is lowered. Right? ये बात समझ आ रही है? कि जैसे ही energy reaction start होता है, आपको एनर्जी कंज्यूम करनी है, तो एनर्जी बढ़ रही है. जैसे ही डिमांड फुलफिल हुई, द रिएक्टेंट्स और सबस्ट्रेट विल ऑटोमेटिकली कन्वर्ट इनटू प्रोडक्ट. और जब सबस्ट्रेट इज कन्वर्टिंग इनटू प्रोडक्ट, एनर्जी इज लोअरिंग. दैट हैपेंस इन एवरी reaction in the world. But what happens is that, look at the pink graph and look at the blue graph. Pink graph is showing energy consumption without enzyme, and blue graph is showing energy consumption with enzyme. I mean, when you haven't used enzyme, the demand of activation energy is too much. Let's say 1000 kilowatt, 1000 kilojoules, right? But when, as soon as you used enzyme in that medium or in the cell or in the bowl, the demand of activation energy is lowered. Look at the blue graph, blue peak. The demand is lowered. And this is happening because of enzymes. Enzymes lower the demand of activation energy, which is like 200 kilojoules, which I told you earlier. So the whole graph is telling you that when you use enzyme, the demand of activation energy is lowered. But when you don't use enzyme, the demand of activation energy is quite high. So that is what the purpose of enzyme is. A reaction will proceed at a very faster rate and less time will be consumed. Less energy consumption, less time consumption. I'm sure you have understood this graph. If you haven't, you can ask me question. I will answer it, right? So now, what happened? Site of enzymes. Okay, so now I'm going to talk about site of enzymes. By this I means there are different places of enzymes. Some of the enzymes are within the cell. Some of the enzymes are working outside the cell, into the extracellular space, outside the cell space. So there are enzymes who work inside the cell. For example, the enzymes of glycolysis working in cytoplasm. I have told you glycolysis before. It is one of the steps of respiration. we have discussed it in mitochondria, that glycolysis of respiration occurs in cytoplasm, while Krebs cycle and electron transport chain occurs in mitochondria. So these series of reactions, which we call glycolysis, occur in cytoplasm. It requires enzymes. I mean, the reactions of glycolysis to proceed, it requires enzymes, and those enzymes are present within the cell. So we call them intracellular enzymes. And there is another category of enzymes which are present outside the cell, like pepsin enzyme working in the stomach cavity. You know, there's a pepsin enzyme which digests protein, and it is released into our cavity of stomach. So the enzymes are released from the cell into the space, into that cavity. This is one of the example of extracellular enzyme. Let's look at the diagram. Intracellular enzymes are present within the cell. Look at the red dots showing the enzymes inside the cell. And look at these red cells, red dots in extracellular enzymes. They are present outside the cell. This is one of the examples showing you extracellular enzymes. So today I'm going to teach you about, this is all about what I'm going to teach you today. Because the next topic is regarding, either I'm going to start lock and key method or induced fit method, or I'm going to teach you characteristics of enzyme. So for today, I feel it's better to focus here, stop here, and recall what I have taught. Okay, what enzymes are, what their, we have discussed about metabolic reactions, what is catabolism, what is anabolism, and remembering these equations, kindly write it down in your book, these reactions, right? And then we covered about the energy transfer, how the catabolism releases energy, how anabolism absorbs energy, and how we call the enzymes as catalyst if they are in a non-living environment. We don't call them enzymes, but if they are in a non-living environment, we call them biocatalysts, and we also call them enzymes. And then we studied that enzymes are protein in nature. They do not change during the reaction. See, girls, I am looking at the highlighted word in my presentation. I am focusing on orange, the bold ones, and the different color. So you can also focus on them when I teach. Then I'm talking about, we have discussed substrates and we have discussed reactants. We call them reactants in chemistry. And these substrates are converted into products. And then we studied the diagrams of ES complex, enzyme substrate complex. And then I've told you about activation energy, how it acts as a barrier, how we overcome this barrier, and what is the function of enzyme. The function of the enzyme is to lower these barriers. Then I told you about this graph, how enzymes are lowering the demand of activation energy. We haven't discussed how, because that is a bit complex. So we will discuss it later on. And then I talked about intracellular enzymes and extracellular enzymes. So this is all for today, right? Allah Hafiz. Hope you understood well, and you can ask me questions. Stop sharing and let's end the recording. Allah Hafiz.